river and enjoying going out. Good one. Thank you, Anya. But we've also always it's quite powerful the amount of data we're collecting now with everyone all around the world. And we this year wanted to explore that a little more and see if we can actually do something with those data. But instead of us as a team doing this for everyone else, we more want this also to be collaborative and see if we all together just can dive into those data. But we realize how big the challenges of doing something with global data that are some spots around the world have some rivers and a lot of spots also not. So it's hard to create something global. So during the session, we want to do two things. One is first looking a bit, taking the time to actually brainstorm a bit about your own data. So we'll make, I think, 3 breakout rooms later. So within smaller groups, you within yourself can discuss a bit and can say a bit during our Bioblitz, we had a lot of those kind of observations, and this is something actually special, and maybe think if we can get something more out of that, no? And then we'll have a discussion more about the global data. And since Andy is here, who was actually going to present yesterday, but she will kick off this whole global view, I think focused on fungi, no? Is that correct, Andy?
And plants, but yeah. I'm sorry, I didn't know I was supposed to present yesterday. I would have been there.
That's not your fault.
Me neither, Andy. Me neither.
Okay, I'm gonna quickly share my screen because I actually prepared this a bit in the presentation, so it might be more a better overview. Which one is it? No, this one. Okay, so yeah, I'm here today with also Anya and Jens are here from the team, but definitely Jelena, who is our science wizard always, and she's yesterday presented quite a bit about the birds already, but always curious about the global data. And maybe Jelena, you want to mention already what you're planning for later this year?
Hi. Yeah, so I had I'm kind of trying to wrap my mind off around this Bioblitz data for some years now, but I never have the time to do that because I have other data to take care of. But now I got invited to give a presentation about biodiversity in one research center in Oulu in Finland, and I thought it would be really nice to actually present something about the Bioblitz and citizen science. And finally, I will have to force myself to look into some data, which is a great opportunity to explore this pattern more on a global scale and actually see what are the pros and cons and what can be done locally and what can be done globally with this kind of data collection. But yeah, so that's going to be something for February, where I will try to present something about the Bioblitzes and all the experiences that people had, and also try to put it in a more scientific context and see how to present this data in a more reproducible manner. But yeah, so that's my plan. We'll see how it goes.
Yeah, cool. Super cool to know that also somebody's really interested in data, right? And we hope that more of you in the group are. So as mentioned today, we want to first exchange a bit on the local experiences on the data side, know what's in there, chat a bit if we can figure something out. Then we're going to explore the worldwide data, and then I want to still have a bit of a brainstorm with you guys if there's some research question that we could actually ask ourselves and that we could answer maybe with the data we're collecting worldwide. So first, this exchanging experience. A few questions maybe we'll put you in breakout rooms, and maybe a few questions that can lead your discussion is okay, did you have before the Bioblitz already any scientific goals? And the data that you collected, were they sufficient to maybe answer those? Was there anything that stood out? And maybe even taking it to okay, there were certain observations that were interesting, but maybe next year we can focus a bit more on this group of species or on this kind of for instance, okay, water quality is a big issue. We want to focus more on macroinvertebrates and understand more which invertebrates tell us something about the water quality, no? So I'm gonna stop sharing now, and I'll put those questions quickly in the chat as well. So you have them close. And then, Anya, if you would want to create some breakout rooms, I think we're maybe even 5, huh? So it's like small groups, and within the small groups you can chat a bit together. Does anyone, before we go into the breakout room? Yeah.
Sorry, I can also just like always go to each breakout room, and maybe if people are interested in actually like going into their project and extracting the data from iNaturalist, I can show them how, and we can also like brainstorm about other things. So yeah.
Yeah, cool. Is there any questions before we go into the breakout room on the idea of this? Is this clear? Cool. I think then we can go, and let's do this for about 10 to 15 minutes. And then we can come back together and we can share a little bit if there's something that came out from this, yeah? Okay, and yeah, go ahead whenever you're ready.
Hey,
Hi.
Paul. Where are you from, Paul?
Hi.
Hey.
Oh, I'm from India.
India. Are you in?
India.
Hello, everyone. How are you?
Hi, hello.
Good morning.
Good morning. Do you have any questions or doubts about what you are going to do here?
¿Respondemos directamente? O sea, nosotros,
Yeah.
yo te respondo en castellano, mi inglés no es muy bueno. Nosotros teníamos sí objetivos científicos en algunos lugares, y si los datos fueron suficientes, no, en realidad. Porque
Okay.
obviamente lo que estamos haciendo es rescatar gente todavía. Estamos enseñando a cómo funcionan estos BioBlitz y cómo incrementar la participación. O sea que, por un lado, eso fue bueno, pero hasta por ahí nomás, digamos, con la ciencia.
Okay, let me share your opinion with others. Benjamin is saying that they had objectives, but since they are working on how to empower the people to use iNaturalist application, there's been like a challenge in order to have more data, or the data that will represent the river in iNaturalist, I mean. Is that correct, Benjamin? Okay. So is there is there anyone that is experiencing the same challenges? Like it's a challenge to upload the data on iNaturalist, or what are your experience on iNaturalist data? Maybe Jane or Michael or Antonio? Yes, Jane.
Okay, so for us, our experience with iNaturalist is not everyone can upload to it, but the few of us can upload to it. The data ain't that much. We just had about 32 observation, but our objective was how well can we teach our young ones, the children on there, getting to know the organism, the insects around and getting to know our rivers around in Kenya. So we are still getting there, still learning some of the insects around. And something that stands out for us is for the children getting to know, like, a moth, it always starts out from the small larvae, which looks the stories were like it's moving like a caterpillar. So they were amazed and happy to see how, and you're still empowering them to get to know more of the insects and how they grow, they develop. Yeah.
Interesting. I see that you in your group, in your home River Bioblitz, there's a lot of kids around it. That's great, and the tools that you're using.
Yeah.
Thanks, Jane. Antonio or
And
Michael?
also,
Yes.
sorry to
Continue.
and also to add, yeah, so we use more of for them to get to know them, we use the foldscopes for them. They're tiny friendly microscope, and also after they view it, we are able now to upload it in iNaturalist, and they get to know the scientific name of it and get to know which family they're in. Yeah, so that's our experience so far.
Oh, nice. Yeah, I saw the pictures where the kids are looking up with the tools. And that's nice because they are more connected to the individual of the species. So that's interesting. So now Antonio, sorry.
Yes, I was planning to, I want to mention about some lessons of this year experience, because we were also participating in the last year event. We have, like Argentina, for example, the same situation with the people. We have a very variable situation because in Peru we had 14 different projects in different rivers with different kinds of groups. We have involved in some ones students, and other ones we have some students of university, not real scientists working in the teams. So the people was more animated, more stimulated to bring participation and not really understanding the real needs for to obtain better data. What I'm trying to meaning is the people was trying to capture all the biodiversity possible, but they don't have the real assistance to know where to find species that are really more relative to the river. I am thinking about biodiversity species that are really indicator for the river quality. But what have one of these groups, one of the 14, with 2 students from one local university that were taking water samples, and after they were making the review in the laboratory, and they have identified several protozoas, and they have this. This is the initial step to teach to the people how to find more species of the water quality importance, no?
That's very interesting. Yes, let's see. Next, Michael, let me just say something about Antonio's mention. Is that actually there's a challenge with citizens and a challenge for scientific data. What we did here in Mexico in 4 creeks is that we went into impacted river, and then we went to a conserved river, seeing like in the vegetation it's more conserved than the other. So that's how we managed to classify observations into maybe good or maybe bad, because we have that challenge of what we are seeing in the river. Okay, Michael.
Thank you so much. Mine is more of like a question because we're working closely together with Janice. So Janice have basically shared a little bit of our experience. My question goes, and this is something that within EduTab we were thinking of which way forward. When you look at the River Bioblitz, home River Bioblitz data, from the iNaturalist, there is a huge, rich chunk amount of the backend data, with of course the privacy all taken care of. To what extent can such kind of data be what are some of the opportunities that kind of data can be used, for example, institutions like EduTab, who are very keen on, number one, looking from the also having sessions to take these participants or children to understand the concept of data and how it informed our ecosystem, and two, even from the research point of view. What are the opportunities about that are available in those two lights?
If I understand, it's how to use the data from the iNaturalist to connect with the citizens, like as a feedback. Well, we have been working on the storytelling, on the infographic and maps that are more visual for the citizens. That's a way we can do a feedback from citizens here in Mexico. What we are doing is doing meetings with citizens and their communities in the communities where the rivers are. So what we are trying to do is a kind of a collage of the map where the citizens were taking the observations, and then a collage of the species and identifying the species that are more interesting. For example, when you enter into the iNaturalist platform, you see all the observations, and you see some of them that are research grade. So in those research grades, you can be sure there are validated from the specialists. So then you can see more about the information, and some of those observations, for example, here in Mexico, some of the observation of the citizens are very rare species, or are species that are under some danger. And those observations, we took them and we put them in presentation or infographic or storytelling. That's how we are trying to reconnect the information with the citizens. And also use social media, or use the platforms where the same platforms where you invite the people to join Home River Bioblitz, the same platform to share what you find as a map or just the photos from the species. The other way around, it's like in the scientific part, we are proposing to use the data as an indicator of quantity. For example, in conservative places that we know that are conserved in the river, we expect more observations than places that are more impacted. But that's like a more analysis effort in that part that could be combined, as Antonio mentioned, with quality data, the water quality data. That's how we can do a merge between quality data from water quality index and the observations of the biodiversity. So will depend on how, first, what will be your audience? If there's the scientific audience, maybe the merge or the analysis between water quality and the iNaturalist observations, but if it's going to be a social audience, infographics, storytelling, and image will be great. I don't know if I respond your concerns.
Yeah, I think you've explained it in a more simplistic way, if I may put, but in case we have more other questions, definitely our team will reach back to you regarding key specific questions. Yes.
Thank you. And with that, I will want to ask you, did you find some species or images in your river in this Home River Bioblitz experiences that you are keen to share to your audience? Like you think it's important to share that information with your audience? For example, we find two really interesting things in Mexico. One, that we find a rare frog that is just in a very good rivers. So we want to use the photo and share, hey, we have this frog, we have to conserve this place of the river. No? And the second one is that we find a really bad situation of a lot of garbage. So with that, we will do another publication saying, hey, we have a lot of garbage and we have a frog that is really rare and we have it. So what kind of really interesting like image or experience did you find in your experience in the river?
I don't see everyone of my group yet, but let's
Mine is
hope.
almost there. Just Paul is missing.
I don't think Elaine is
Comment on what they talked about in the breakout room. Something that like stood out. You're like, oh, this was cool. This is the research we want to do.
maybe I'll just make a quick comment about the group that I was in. It seemed that the main goal for them, for most people, was to get like young kids interested and involved in their like local area and like getting them interested in the plants that are growing around the rivers and you know, having them feel connected and also like learning a bit. I thought that was really cool.
Nice. Thanks for sharing, Andy. Hey, Elena, you've been jumping around quite some breakout rooms, so maybe you have a bit of an observation.
I actually didn't manage to go to many. I just went to one and then I went to yours and then we kind of finished because there was not so much time, but sorry. But what I was really interesting talking with Lazarus also because they are trying to figure out kind of like a question for next year and what to do with all this, what they've been collecting. They also said they've been collecting some data that is from two different spots that are differently impacted with different biodiversity. And I think that's a good place to start with everybody as well. If you have some spots in your river that you think are particularly interesting because of the human impact there or because of some biodiversity there, you should go and explore. You can always make a comparison with both spots. You can go to them between years, 1 year, one spot, the other year, another. Ideally, in the same year, you should probably do both. And yeah, so I think that's kind of like, I see there's like a potential that they could check these two spots. It's also really good to, if you did a bit BioBlitz, to do it at the same spot. So to not really mix, to not always go to a different place and randomly choose it. So if you want to have some, like if you want to do it for a reason scientifically, if you want to prove something, if you want to prove that this species is there year by year. For example, this year we had some endangered bird that was previously years, I think, at the same river, which was pretty cool then, because these species that are very charismatic and like they're kind of flagship or they are threatened or endangered, it's a really good way to kind of help you promote your river and also protect it in some way. And not only because there's this bird, but I'm sure there's a lot of other species on this spot. And if you can prove that they are occurring there continuously, it might be a very good argument for protection of the river or the part of the river. Yeah.
Well, thanks, Elena. Is there somebody else that wants to share something with the whole group about that conversation they had? No need. I remember one thing from actually yesterday, the recap session that got my attention a bit, which was in Mexico, I think the Salas River, but I'm not sure. That they commented that they saw that this year there were a lot less insects all around than the other years, because they've already done it a few years now, so they've already seen a bit of patterns compared to what was there around this time of the year last year, which was cool, because then they already said like, hopefully it's just because the rainy season is starting a bit later this year, so there's like less rain, so less insects, less flowers, everything. No, maybe it's also something worse. It could also be contamination of the river or something, but it's a cool, if you do it every year you can start to see patterns, and then there's also a reason to start relating that to maybe what happened or natural disasters or whatever. No, and it could actually make you more aware also of like human induced disasters that are happening. Cool. That was, I hope, a bit helpful for everyone to like look into their own data. Sorry, Anya.
Yes, I want to hear about the thoughts in the group that I was. The first, there was some challenges mentioned. The first one is that there were a challenge on how citizens use and upload the data. That it's kind of a ongoing process between uploading the data, understanding the data. And the second challenge is that sometimes people are uploading the data, but doesn't know what are they looking. Like, is it a good sign? Is it a bad sign? And the third one, it's a concern on how to use or how to understand the data and present it into a social environment. That's the third one. We were into the questions in the process to the question when everything closed. But just to mention, here in Mexico, we found a frog in the upper part of a stream that it's very interesting, while in the down part of the stream is very impacted and the frog is not there. So it's really interesting, this change, just to mention.
Cool, thanks, Anya. Yeah, I'm aware it's probably little time to really go through all the projects and to figure it out, but I hope those questions help you to actually think a bit more about your data. Maybe there's indeed a certain frog that tells a lot about the water quality, and next year you tell all the participants to specifically also look for that frog, no? Like, give them a bit of a challenge in a certain direction, because we know that this tells something special. There in my group there was also somebody that definitely mentioned, same as you, Andy, like it's also just a goal to make people aware and to explore, and we definitely don't want to necessarily lose that. No, it's always this balance, like how much do we want to make it science, or do we want to just make it a cool day, and that's up to everyone themselves. No, cool, thanks a lot. Hey, the next thing I want to do is indeed to look a bit more at the world, at the global data. And let's kick that off with Andy presenting about fungi and plants.
Yeah, okay. Hi, everybody. I'm sorry that I was not there yesterday, but yeah, I'll give my presentation now and give you guys like a bit of an overview on sort of the plants and stuff that everybody recorded during their bioblitzes. So let me just, there we go. Now I can see better. Okay, can you guys all see? Is it full screen? No. There we go. Okay. Great. So
Sorry, it's not full screen.
Oh, it's
there
not?
it is. Now it's
Good now? Okay. Okay, yeah. Hi. So I'm Andy. A few of you know me, but for those of you who don't, I'm a PhD candidate at the LMU. That's the Ludwig Maximilians University in Munich, Germany. And I have a degree in botany, and I'm currently studying lichens and evolutionary biology. So this is a bit of a passion and profession interest for me. So yeah, thanks for letting me give you a bit of an overview. Okay, so first, I just wanted to generally compare some of the numbers that we have from this year with what was documented from last year. And what you can see is that with the plants, there seems to be an overall decrease in the number of observations that we documented. But with the fungi and lichens, there's a general increase. So that is, I think, really interesting for me, because usually it's the fungi and lichens which kind of get overlooked or not considered in stuff like this, because plants, there are so many and they're so easy to like identify and take a picture of, whereas lichens and fungi tend to be a bit smaller, or there's fewer of them around. So yeah. And then I just wanted to make a quick note about the ratio of what has been successfully identified and what is still in need of identification or of confirmation of identification. And it's over half of the plants still require that second agreeing identification from another person. And almost all of the fungi and all of the slime molds that were identified haven't been confirmed yet. So if you have any expertise in any of these fields, just going through and looking at some of the uploads from the different locations would be super helpful, because that makes, then if you can get a good ID on those individuals, then you can bump that quality up to research grade, which means that it's like a confirmed identification rather than a suspicious identification. Yeah, so if you can help, please help. For example, this is one of the identifications that was documented in Mexico. And I got really excited when preparing this and looking at it, because Zamia is kind of an endangered, threatened species. And I was really excited to see that someone had found it. But looking closer at the photograph, I noticed that it's actually not. So Zamia is a cycad, and it's like a native plant in Mexico. And you can see it there on the right. And what was documented was an arundo, which is actually a super invasive, aggressively developer of monotypic stands in riparian systems, and they actually lead to habitat destruction and like biodiversity degradation, because they sort of just spread and make these sort of monotypic stands. So this is sort of a two-part thing, where if we really could use help with confirming these identifications, and then because then we get like a realistic idea of what is actually in these areas, and then we also can learn, you know, what are these species that we have in our riparian areas. Yeah, so this is just a quick overview. Everyone who participated in the Bioblitz documented plants, which is quite obvious, because they're easy to photograph, they don't move, and they're easy to get into focus. We had kind of a high percentage of what could be considered introduced or invasive species in our data set. So almost 200 of the species that were noticed are considered introduced or invasive, including the arundo that I mentioned. And only 2% of the species that were documented are considered of threatened or endangered status. So I think it'd be really cool if we could somehow bump up those numbers of the threatened species, because it's good to know where these species are located, and then we can, you know, sort of encourage our local governments to protect those areas more if we can show that there are threatened species in those areas and they deserve protection. So yeah, for example, these are two of our most observed plants that I found in the iNaturalist dataset. One of which is a quite notorious invasive plant called the castor bean. And it was actually found in quite a few of the different bioblitzes on different continents. And this guy is pretty pernicious. He actually can be really toxic if ingested or even touched if you have a latex allergy. So it was originally introduced as like an ornamental like plant. People thought it was pretty and wanted it in their yards and stuff. And then it sort of just has spread and you know, can be quite dangerous for animals and children because if you eat too many of the seeds, they're actually toxic and can be lethal. So I know where I grew up in California, there's some initiatives where people go in and actually physically remove big stands of these things to try to help the riparian areas recover. So I don't know if any of you are involved in things like that, that would be a good plant to target. And then one of the other most observed plants I thought was really cool is a really beautiful native tree, native cypress tree that is native to Mexico. And yeah, I love cypress trees. I think they're really great. And it's nice to see so many of them documented in so many different locations. So I tried to pick out a couple really beautiful plants as well from some of the different locations. This is one that I thought was really nice, another native plant to the Mexico, to the riparian areas, and it has these beautiful vanilla scented flowers. I think it'd be so cool to see these in person. And then this is one of the plants that we saw on our trip in Munich. And I thought it would be a cool one to sort of do some more detailed information on. This is a Himalayan knotweed or Himalayan balsam. And it's another really aggressive invasive plant that was originally planted as a ornamental and sort of has just spread and taken over, especially in riparian areas where it's done a lot of damage because it outcompetes the native riparian plants. And when it dies back in the winter time, it sort of really promotes riverbank erosion due to like the integration of the water and stuff. So yeah, it's pretty awful invasive plant. But what we talked about in Munich, since we were discussing useful plants or edible plants or medicinal plants, is this one is one of the edible plants. So it's in some places people are using its edibility as a way to cut back on how much is around. And you can eat the young shoots if you cook them, and they taste similar to rhubarb. You can eat the seeds. They have like a nutty taste. You can take the whole pods and cook them and eat them kind of like sweet green peas. The flowers are edible. They can be used in salads or for dyeing drinks like lemonade or something. And so eating this plant is maybe a good way to remove some of it from the environment and reduce its spread. Just be really careful not to accidentally spread it yourself as you're trying to harvest some of it or anything. Yeah. Additionally, so this is a a cucumber relative that is native to the Southeast Asia. This is one that was documented in Thailand during their meetup. And I had never heard of this guy before. He's really cute. Makes these tiny little gourds, and apparently they're edible. Not the gourds, but the leaves and the stems. So maybe if someone has some information on this guy, then you could inform us a little bit more about this one. But yeah, I thought this was a really cool find. And then also with the BioBlitz that happened in Tanzania, I was trying to go through a lot of the data, and I noticed that there's just so many that hasn't been identified. And if anybody is good with African plants, if you could help us get some solid IDs on these, that would be really helpful, because there are some really beautiful and interesting specimens that I don't know personally, and I couldn't really pull up any good information on because of that. But you know, there's just a lot of diversity, and it would be really cool if we could help them get some identifications on these and start helping build up their data set. Okay, so I'm going to move into the fungi really quick. Let's see. So again, almost everyone documented at least one fungus, which is very cool because, again, normally they're a bit overlooked. So these were two of the most observed fungi that we're seeing. You know, both of these are involved in different types of nutrient cycling. So either like decomposition of wood or other detritus. And they're really important parts of forest or riparian habitats as part of the nutrient cycles. So it's really cool to see these. And then again, staying in the theme of things that are useful, I found that there were some documents of an edible fungus. And I think this is one that's good to talk about because it's super easy to identify. So it's good for beginners. If you don't know a lot about fungus, this is a good one that it's really hard to tell. Like there's no real poisonous lookalikes, and so it's a good one if you want to start testing yourself. But this is Lycoperdon perlatum, and it's called the puffball. Identifying features are this very spiky textured surface, where if you just touch it, it sort of can just crumble right off. And it has a very small stem, which is dense with fiber. And if you cut them in half, they're pure white when they're young, and that's when you can actually eat them. And I think they have a quite strong flavor, very strong mushroomy flavor, so not everyone would like them, but yeah, I think they grow almost everywhere, and they're really easy to identify, so it would be a good starting point. A couple interesting fungi that I saw were these amorphous bird's nests, which I'm not sure. They kind of look like a dog came over and puked up some lentils or something, but these are actually the spore packets, so that's how they reproduce. It's a really strange system that only a few fungi have developed, but I think these guys are really, really cool, really unique. You know, I would say probably not edible, I'm not sure, but probably would go against that. And then this other Entomothera, this guy is super cool. Actually, so what it does is it parasitizes insects, and you can see here where the red arrow is pointing. The fungus is actually growing up through the different segments of the fly exoskeleton. And what it does is when the spores land on the insect, it sprouts and grows inside until it takes over the whole body and and the fly dies. So it's some of these types of fungi are used actually in pest control, but yeah, I think these guys are just really, really interesting. And then because they're my favorite, I wanted to highlight a couple lichens because they so often get overlooked. But these were two of the most observed lichens. I thought it was really cool that this Xantha Mendoza was found because it was so tiny, and someone got a really nice picture of it and also was able to identify it. And then finally, because these guys get looked over even more than regular fungi do, this is one of the cool slime molds that were found. So the picture on the left is from the Bioblitz, and then on the right, these are just some examples of what it looks like at different stages, different angles. But basically, it's kind of like a fungus, but not quite. It's a little bit more complicated. Ask me for details if you're interested. But yeah, anyways, I think I've talked about enough. But yeah, thank everybody for participating in the Bioblitz and for letting me come and talk to you about the things that I enjoy.
Amazing. Thank you.
Amazing
Andrew.
review.
This is so cool. I'm really impressed with all that you figured out from there. One question already from my side. Everyone that wants to ask questions, feel free. But because I'm not that into our naturalist yet, still. How do you like figure out which species are introduced? Does it have that label or something?
Yeah, so actually this is a cool function that I noticed. Maybe Yelena can show us or something, but in the if you go to the Bioblitz like page for everybody, there's a little section that says filter when you're looking at all the species, and there's a bunch of different options for, you can filter for animals or for plants or for fungi. And then there's also another section where you can filter for things that need identification, things that have been confirmed, identified, things that are research grade, things that are introduced, things that are endangered, all these different tabs or options that you can filter for. So I think they do a pretty good job of keeping up on the like where this species was documented versus where maybe it's native, and they only mark the ones if they're documented outside that zone as the invasive ones. It's really cool.
Yeah, I can show you how to extract this, like by sharing my screen.
Yeah, please.
Yeah, so this is indeed, yeah, please, Yelena, because this is indeed exactly what we want to do now with everyone. I'm gonna also quickly share you the link of the global project now, to just explore a bit all together, so we can go to the observations of this home River Bioblitz, and then do some filters or look at specific species. I, for instance, looked up kingfisher, and I was like, oh, there's actually 34 observations of kingfishers all around the world, and it's 5 different species. I was like, oh, that's cool, because kingfisher is such a icon species of the river. So yeah, if Yelena, maybe you want to show this a little bit, what's all the functions, and then everyone can just take 10 minutes or something to see something that they find interesting, right?
Yeah, that sounds good. So you can just, when you go to iNaturalist, you can actually access any Bioblitz for any year that was in a project just by typing the home River Bioblitz and then a year. So there you see there's 2023, there's 2021, 22, and then once you go in the home River Bioblitz, so in the project, you can click about. You can see the different projects from different rivers here. So you can always access your own data from the river, or you can you can check out the global data. I can, for example, so there are some stats here. you can always see how many observations were there, how many species, how many identifiers and observers. And if you click on this stats, then you can get some statistics about, you see, for example, here for the whole BioBlitz, we had 12,000, almost more than 12,000 observations, but a lot actually still needs identification. As you can see, that's like more than half, and only 4,000 are with the research grade. If you look, so when you just hover over these graphs, you'll see this information. And here you get different species groups, and you can access them by simply clicking on this. So I can just go to birds, let's say. I'll click on this blue part, and then you can see which bioblitzes have recorded birds on this map here. Here you can even like hover around and then see the locations of birds that are kind of coming here with the pictures. And let me just see. So now I have only chosen birds. You can choose anything else. Again, something here about birds, how many species, how many identifiers and observers, and then what Andy was talking about, you click here in the filter, and here you can filter for many things. It's always like you can click first what has the research grade. You can also filter what needs ID, which species are threatened, which are introduced. I've never clicked on popular, but okay, apparently it has one or more faves. You can also filter for that. You can also search, for example, yesterday I searched hummingbirds aggressively with all caps. And then it will hummingbirds, and it should give you then the results of only hummingbirds that have been observed globally. So you see here there's 46 observations, 9 species, and 9 identifiers. And you can see that on the map, or if you go into the species, then you really see which species are those. So these are not the photos from the observations, but if you click on this 9 observations here, then you'll be led to the proper observations. So for example, like this, and then you see that this species had 9 observations, this ruby-throated hummingbird, and they were in these spots here. You can always zoom into the map and then kind of see in a better resolution where they were occurring. That's really interesting. And then what you also can do, so you can just remove this. There's a lot of, for example, for threatened species, you can take that and then you update your search. And we should be led to the threatened species of birds, but
you have to X out that.
I have to go here and then yes. So this is now just species for this for this region, I think here that are, or no, these are all species. Okay, well, I have to go, I'll go back here so that I don't click back too much. So if I go again into birds, then I can filter here for threatened species and I just update the search. You can do many things. You can also like, if you know somebody with the username, or if you want to see your inputs, you can type your username here. Or if you're interested in specific place, you can also put the place here, and also the dates. For example, you can put here the range of dates of when BioBlitz happened, but you don't have to do that because this project is an umbrella project and it's set up for these particular dates anyways. So if you update your search, then let's see. There were
I don't want to interrupt too much, but maybe it's because we're also don't wanting to make this session too long. Maybe it's time to actually go into the observations.
Yeah. So this is okay. So do we go now? I can stop sharing and then everybody can explore what they want, or okay.
That would be great. And I'm gonna quickly start this whiteboard So so the idea is like with the information that we now gather, everybody finds some interesting stuff. It doesn't have to be super high scientific knowledge or something, just something that catches your attention. Let us know and just put it on this whiteboard, and later you can just put a post-it or whatever. Later we'll try to make an infographic from this. So it will be an infographic with the results of this hackathon now. So let's take about 10 minutes to just go to this link of the Home River Bioblitz of this year and use those filters, zoom into maybe only a certain continent or something, or whatever you find interesting, and anything that catches your attention, put it on a post-it, put it in some text, and just add it to the whiteboard. Now our challenge is to keep silent for a while so we don't disturb people.
Sorry, what are we supposed to do? Right.
Yeah, you still hear me here? To go to this link that I just sent in the chat, no? Which is the umbrella project. And to just go explore. Maybe you're interested in plants and you filter on plants. Maybe you're like trying to see if there's invasive species. Yeah, just take 10 minutes to kind of explore all the data that were collected worldwide. Maybe somebody wants to compare stuff that was found in Africa to stuff that was found in North America or something. Whichever gets your interest.
I was trying to be lazy and I took a screenshot of the thing I wanted to share, but it's not letting me post a picture. So I'll have to type it out.
Did I just do this again?
Yes.
Oh, that was on a different device. Okay, there was not much in there yet, no? I'm just super. The audio on my My computer's not working anymore, so I'm also on my phone, and I was trying to mute myself, but I couldn't find it. I'm so sorry. Is everyone enjoying the exploring, or is somebody really lost and needs a bit of guidance? I think everyone is enjoying exploring. Just if somebody's not able to really use that whiteboard, also feel free to just put something in the chat that you would like to see there. Okay, we'll keep this going for another 5 minutes, just as a remark, to try and look at the global data at this moment, so of the whole home River Bioblitz umbrella project, rather than at specific rivers. Yeah, I think it's time to slowly round this up. In the meantime, I'm gonna just ask a few questions on the comments that were made. So for instance, the one that says 32 different plant and flower species, with a mile a minute vine in the Leonard's Kaya. Who put this? Is there somebody here that like wrote this down? Because I'm assuming this is specifically for one river, no? Nobody there. Okay, maybe one that is for
If
sure
you
about the
can,
roadblock.
if you click on it, you can click the little 3 dots next to, and then there's the info tab, and it tells you who made it.
Oh wow. Where is, wait, I have the 3 dots. Ah, info. Ah, Victor. And he made it 3 minutes ago, so I'm assuming he's still there. Victor, are you with us? Okay. So then Paul, Are you with us? Because you made this one of that the 40% of the bird observations are passerines, lower than a global proportion of 60% due to the overrepresentation of wetlands-associated non-passerines. Can you explain this one to me? What are passerines?
Oh, you're talking about the passerine proportion, right?
Hmm?
Wait, are you talking about the passerines? That is mine.
Yeah.
Oh, yeah,
I'm
so
like not a birder. I don't even know what passerines are. So.
oh, as in songbird representation in the River Bioblitz is as expected, because there are mostly non-passerines when you come to wetland associated habitats, right? Because you have a lot of storks, pelicans, kingfishers. So these are all different families which do not belong to the songbird groups, right? So that's why we are getting. If you're taking a bioblitz in different habitats, say grassland or a rainforest, you'll probably get a lot more passerines compared to non-passerines. That is songbirds compared to non-songbirds.
Cool. That's a cool observation, no? And it makes sense with us doing the home river bioblitz now, and not the home grassland bioblitz or the home mountain bioblitz as now. Let's see. Cool. Is there somebody that wants to specifically zoom into their, that wants to explain their information they put? One question to Antonio. Are you with us? You did a note on the chironomids. I think that was also specifically on the Lorraine River, no? Okay, guys. This all was a bit of an experiment. I think I'll check with Jelena later, like what information from this we can gather again, and we can make this potentially into an infographic. If you're enjoying this process and you're still looking into the data, feel free to send some information still also on the WhatsApp group or something. And in the meantime, I'm gonna close this spreadsheet now, close this whiteboard. Should be saving. If there was something you were just writing down, also feel free to put it even in this chat. But for this moment, I'm gonna close it so we can see each other again. Hope that's okay.
It was really neat to see all of the things that people noted down.
Right?
Yeah, it was fun. It looked like it was fun.
I also liked it. I was playing around looking at snakes and herons and egrets.
Yeah, it's quite fun though, to take a bit of time to actually zoom into all the data and focus and stuff. Amazing. Hey, one last thing I wanted to do during the session, but I think we're actually kind of running out of time, is think a bit of like, okay, now we've looked at those global data, we've seen what's happening around the world a bit, and we always have this question in the back of our heads: is there something we can do with those data? Now, is there something that we can really say about our rivers globally? That is based on the data that we do with the Home River Bioblitz. So is there like one research question that we can ask ourselves globally that we can answer with the data that we have now? If there's somebody now that has an idea in mind, please feel free to say it. Otherwise, have this in mind, and if you ever have an idea, we're always in search for this kind of question, and feel free to contact us, and maybe we can work on it a bit for next year. Yeah, in the meantime, I think we'll close this session, and that means also we're closing the sessions of the whole Home River Bioblitz of this year. And clearly this was the 4th edition. That means that next year, we're obviously doing it next year again. I would say no. Jens, do you agree? There's too many people already excited about next year. So the 5th edition, if somebody is like, oh, we should do this for the 5th edition, that's going to be super cool. Also feel free to write us. We're always open to ideas. And then it's going to be a few months, and then we'll start or half a year at least, we'll start activating the chat a bit more. You'll hear from us still in those weeks. Also keep uploading your photos on the drive, as Anya has always been asking, so we can use them for the video and for the social media in the coming time. Thanks a lot for joining this session. This was cool. As said, it was a bit of an experiment, but I think we got some good information from all of you together. Jelena, do you have some final words on this session?
Can make up some final words. I really enjoyed everything. It was always it's always super fun to go through all these observations after everybody's finished with their bioblitzes and listening to the presentations. And I really enjoyed Andy's presentation about plants. I think it was really informative. And fungi and lichens. And I really hope to see all of you next year. And don't be afraid to go into the iNaturalist and click on the stats tab and extract your data and play with it and make your infographics. And if you see something really interesting and useful, grab it, then do it next year. Focus on one species that you really want to, or just do like an informative bioblitz for children. It's like create your own event. But if you need any help with creating a more scientific protocol, you can always text me or reach me if you want to brainstorm ideas. And I think you all also saw Enya's presentation that they are having this really cool idea of bridging citizen science with regular science, with other type of science, the science. So yeah, I think this bioblitz can be really an inspiration to explore your rivers, and especially if you want to conserve them, please check the biodiversity, get in contact with experts and people that can help you, that you can collaborate with, that can give you an approximation on numbers of the species and and estimation of the biodiversity in your area, so that can help you protect your rivers. Yeah. So
amazing. Glad I asked you for some final words. Those were great.
Cool.
Okay, I think with that we can then close this session. Thanks a lot, everyone. Thanks, Andy. Thanks, Jens. Thanks, Jelena. And Anya.
Thanks, everybody.
Thank you so much.
Thank you.
Thank you. It was nice playing with data.
Yeah, thank you.
Cheers.
Bye.
Thank you. Bye.
Bye.
Bye.